At the onset, let me thank all the organizers of this wonderful conference, the International Cancer Conference 2023. My name is Jeevan Ghosalkar. I am a part of the Discovery Biology team at the Cipla R&D Center located in Mumbai. Just to give a brief introduction about Cipla. So Cipla is one of the largest pharmaceutical companies based out of India, headquartered in Mumbai. And though it's one of the largest generic organization involved in generic drug development, it also has aspirations in the field of basic research. And the work that I'm going to present today is a part of that strategy that Cipla as an organization has to conduct basic research. And we have conducted this work in the field of drug repurposing as a part of that project. So the title of my presentation is Ketorolac modulates HIF1 alpha DDX3 RAC1 beta-catenin axis via a novel tumor suppressor prostate apoptosis response 4 against renal cell carcinoma. So these are my declarations. Just wanted to share that the entire work that I'm presenting today is completely funded by my base organization, which is Cipla Ltd. This would be the course of presentation that I'm going to take everyone through. I'm going to first talk about drug repurposing. I'm sure some of you would be aware of what is drug repurposing, why repurposing has gained popularity in the recent past, and as Cipla, what strategy we used while repurposing drugs against renal cell carcinoma. Then I'm going to talk about specifically Ketorolac against renal cell carcinoma. How did we identify Ketorolac against renal cell carcinoma? What is the new mechanism of action that we have elucidated for Ketorolac against renal cell carcinoma? Followed by summary of the presentation and what probable future direction could be for Ketorolac or for any repurposed drug for that matter once you have completed the initial in vitro and in vivo screening of the drug to be repurposed. Now, just to quickly talk about drug repurposing and what is drug repurposing. Drug repurposing, or simply drug repositioning or rediscovery or reprofiling, is a strategy that is nowadays being widely used to find out newer indication for already approved or pre-approved medications. Right? Now, it may not be always drugs which are approved or pre-approved indication. Repurposing can also be conducted for those drugs which probably would have failed at a later stage of clinical development, specifically with respect to their low or no efficacy at all. We would not consider those drugs which have failed due to safety reason, but there is a possibility that certain organizations, certain institutions may also look at those drugs which would have failed as a result of efficacy for a given indication, and they would want to repurpose now this drug for another indication. One of the most recent example that we can talk about is remdesivir. Now, remdesivir was developed by Gilead for some other indication, but then come COVID, and they were quickly able to show the entire efficacy profile of remdesivir against COVID, and then rest is history. So that is what, in general, very high-level idea of what is repurposing or reprofiling or repositioning of a drug. Now, what can be the probable advantages of repositioning against the de novo drug development? These are some of the points that I have listed down over here. Everyone is very well aware that de novo drug development is extremely cost exhaustive and time consuming. Organizations, industries, institutions can spend upwards of $2 billion for the de novo drug development. Plus, it is extensively low on return of investment. Only 1 out of thousands of drugs that are being assessed sees the end of the tunnel, the light of the day, and is approved for clinical use. It takes upwards of 10 years for de novo drug development. As opposed to new drug discovery, the repurposing of a drug is comparatively less time consuming, comparatively less cost exhaustive, and also it is relatively less risky. The pictorial representation on the right-hand side of the slide clearly shows that if an organization is developing a new chemical entity or a new biological entity to be used against a new target for a given indication showing a particular pathophysiological condition, that is the riskiest. The red box over there talks about what I'm saying right now. On the other hand, if I have a repurposed drug which is targeted against a known biomarker for a given disease, that is the least riskiest proposition for an institution. Least riskiest with respect to the outcome of the research, the efficacy, as well as the toxicity profile of a drug. Given these straightforward advantages of drug repurposing, it has been one of the major research area across the world pre and post-COVID, more so post-COVID, because we have seen what a repurposed drug can do, how quickly it can be repurposed, reprogrammed for a given indication in case of an emergency-like situation, which happened recently in terms of the pandemic. Given this advantages of drug repurposing, Cipla as an industry, we as an organization decided that we would also have a look at repurposing to find out new indication of already existing or pre-approved drugs. There are two major steps in the drug repurposing that we as an organization thought. The first step is to identify what is the therapeutic area, what is the disease area in which we need to work on. And the second step majorly is to then decide on or hone on to which drug to be repurposed against the chosen therapeutic area. Now, this slide over here talks about what could be the filters that the organization can use to identify which disease area to work in. And we decided that these could be the 4 filters that we could use to target what disease area to work into. For example, market research, where we need to look at what's the prevalence of the disease, whether the prevalence is very high or whether it is an orphan indication. Now, again, as an organization, the decision depends whether you really want to work in an orphan disease area because there is an exclusivity term that is associated post-approval. So the decision solely depends upon whether you want to go for a disease which is very highly prevalent across the globe or within a particular geographical area, or it's an orphan indication. Are there any gaps or unmet medical needs for that particular indication against which we want to repurpose a given drug? What's the standard of care that is available currently? What is the generic scenario that is currently existing for that particular indication or the drugs that are currently used for that particular indication? What is the patent scenario in that particular indication? What's the pathophysiology of the disease? How much is the complexity of the disease? Whether there is any potential at all for new discovery in that given disease area. If we can use any kind of preclinical strategy for proof of concept presentation. Is there any feasibility of developing better formulation than the existing ones? What could be the potential route of administration? What could be the dosage? And is there a method of anti-generic strategy that can be applied in order to gain some amount of business advantage? So some of these filters were used by us in order to identify the disease area that we wanted to work on. And one of the outcomes of that was that we would like to work in the field of renal cell carcinoma, given the relatively lower rate of survival in case of a metastasis condition. After the first stage, once we have identified what disease we want to work on, then comes the next stage on identifying amongst all the pre-approved drugs, which could be the top drugs, which could be repurposed for renal cell carcinoma or for the indication chosen post this filtering is done. Again, we used multi-pronged approach to hone down to top drugs, which could be repurposed against renal cell carcinoma. So we use these different strategies, starting from disease enrichment strategy, where we specifically look at each and every pathophysiological aspect of the disease. what is the, the pathophysiological conditions that are getting altered in a given disease versus the normal conditions. And the current drugs which are being used for that particular condition, how they are making a difference to the pathophysiological conditions of the disease. Whether the current drug that is being used are just providing symptomatic relief, or whether these drugs are indeed able to reverse back the disease condition. Now, that is a very, difficult job, but then whether they are drugs, they are doing that. We also looked at the pathway enrichment strategy, where we looked at what are the specific pathways or markers that are getting differentially regulated during that disease, and whether these pathways can therefore be modulated for symptomatic relief or for reversal of the disease. We then looked at, now this is not sequential analysis that we are doing. Most of these analyses would go on in parallel. Some may go sequential, but most of them would go on in parallel. Like, for example, sequential analysis would be specifically talking about the 2D structural assessment and 3D structural assessment, where we compare a pre-approved drug against the existing drug used for the indication. And we try to find out if there is any kind of 2D or 3D structural similarity of the drug that we want to repurpose against the drug that is currently being used for the treatment of a given indication. The better similarity would be an indication that the drug would be able to be as efficacious as the current drug that is used for the indication. So 2D and 3D structural similarities goes in sequence. Those drugs which are identified top drug in the 2D structural assessment would move on to the second level of 3D structural assessment. We also do huge amount of data scrutinizing, data mining. I try to identify the putative targets against which the repurposed drug can be used for, whether the target can be a new target or whether the target can be an existing target. So using such kind of in silico, and let me tell you, all these approaches are in silico AI, MI learning approaches that we have used in order to boil down to a given set of drugs amongst all the pre-approved drugs. So we did a blanket screening across the 2,000, 3,000 drugs that have been approved by the regulatory authorities for different indications. It's not that amongst the approved drugs, we have just used a handful of drugs to find out which could be the top for the repurposing process. So it was a blanket screening. So we screened more than 2,500 drugs for each of the parameters that are listed over here, and then boiled down to the top drugs which could be repurposed for a given indication. In this case, it is renal cell carcinoma. Overall, this is the path that we followed to come up to a level that we are today. So starting from the left-hand side, we identified which was the disease area that we wanted to work on, whether it was neurological diseases, oncology, infectives, virology, cardiology, pulmonary indication, so on and so forth. So the first step for any organization as well as us was to identify what was the disease area that we wanted to work on. Once we identified the disease area that we wanted to work on, we did a preliminary screening in terms of the IP scenario. We did a AI, artificial intelligence and machine learning approach in silico analysis to identify the top drug amongst all the pre-approved drugs which could be repurposed for renal cell carcinoma. Once we identify drug and we think that the drug can be successfully repurposed, the first thing that we should do is we should have a provisional patent file. That's what we did. If an organization has the capacity to conduct preclinical proof of concept studies internally, then of course those studies have to be conducted. If not, then the collaborative research has to be done where the studies in terms of preclinical proof of concept studies can be conducted. Are there any biochemical tests that are available to prove the efficacy of a drug against the repurposed indication? What about the toxicity data of the drug that we are trying to repurpose? Then we conduct. Followed by this, we conduct the in vivo efficacy studies. Once we have positive data for the in vitro biochemical testing, in vitro cell-based assays, in vivo animal efficacy, we take we form a scientific advisory committee, we share opinions, we take opinions, we modify our process. And once the entire package till the in vivo animal-based efficacy study is ready, we take it ahead into the clinic, or we partner with a suitable licensing partner in order to take the drug ahead. So this is the path that we have followed as an organization when we ventured into the field of repurposing. Now, so this was the background information that I want to give an idea on how we have gone ahead and worked in this field of repurposing. Now, coming to the actual candidate that we have identified against renal cell carcinoma, which is Ketorolac. And we used all the approaches that I mentioned earlier, the structure activity based relationship based on the ligand property, active target site binding properties, docking processes, disease enrichment, structural enrichment pathways, literature mining. So we followed each and every approach that I had enlisted earlier and then boiled down to this particular drug, Ketorolac, which could have potential in the area of renal cell carcinoma. Now, Ketorolac is a first-generation FDA-approved NSAID for short-term management of moderate to severe acute pain, post-operative conditions. Important point to note is that Ketorolac, and I'm going to come back to this at the end of the presentation, that Ketorolac is a chiral molecule and it is administered as 1:1 racemate mixture of the S-Ketorolac and R-Ketorolac, the enantiomeric mixture. Now, the S form of Ketorolac is very well known as a COX-2 inhibitor with an IC50 value of 0.9 micromolar, whereas the R form is known to be inhibiting RAC1 and CDC42 with the respective IC50 values as noted on the slide over here. Inhibition of COX-2 is known to produce the actual mechanism on the pre-approved indication, that is the analgesic and anti-inflammatory activity, whereas the R form, being a RAC1 and the CDC42 inhibitors, would inhibit cell addition, cell migration, and cell invasion. Inflammation, adhesion, migration, invasion being also specific pathophysiological condition that occurs in cancer. Therefore, we have firm faith that Ketorolac would be able to bring about inhibition of all these activities and be beneficial for cancer. And therefore, we chose Ketorolac as one of our potential candidates. So what kind of preclinical studies that we conducted in order to prove our point? So we conducted in vitro 2D cell-based assays, 3D cell-based assays. We did in vitro 3D combination studies with the existing standard of care, and we took sunitinib and sorafenib as our existing standard of care for renal cell carcinoma. We also elucidated a novel new mechanism of action for Ketorolac against renal cell carcinoma, or let me say Ketorolac against solid cancers. And we also finally looked at the ability of Ketorolac alone as well as Ketorolac in combination with sunitinib to bring about efficacy in an in vivo xenograft mice model. This is the first set of data, which is the in vitro profiling of Ketorolac, and we didn't use one single cell line or two single cell line. We used 11 different cell lines from ATCC as well as PDX model cell line to look at the efficacy of Ketorolac to inhibit the proliferative activity of renal cancer cell lines. The blue graph over here represents the 2D in vitro assay, whereas the red graph represents the 3D in vitro assays that we've conducted. And as you can see, Ketorolac has better efficacy in a 3D model. Now, 3D model assumes significance because 3D models more resemble the actual in vivo scenario. And we are of the firm opinion that in a 3D model, if you can see, for example, let's say A498 data over here, it's clearly indicative that the best efficacy of Ketorolac can be seen in this model with an IC50 of 280 micromolar. And that too in a 3D model. Along with this, we also conducted a 3D in vitro combination studies. So the tabular representation on the right-hand side over here talks about the ability of Ketorolac to inhibit RXL1183 cell line, this is the PDX cell line, and A498 cell line in combination with sunitinib, and we found out that this combination produces an additive and synergistic effect respectively. Similarly, we did a combination studies of ketorolac with sorafenib, and as you can see, against multitude of cell line, it is giving either synergistic effect or an additive effect. So the in vitro profiling clearly indicates that ketorolac would be efficacious alone as well as in combination with sorafenib or sunitinib being standard of care for RCC. Now, as I told earlier, one of the main problems with RCC is the poor survival in case of metastatic condition. So therefore, we did these simple migration inhibition assays in combination with sunitinib as well as alone, and the picture on the right hand side top corner shows that ketorolac alone as well as in combination with sunitinib is able to inhibit migration of A498 cell lines substantially and significantly. Along with that, we also looked at important markers like VEGF, cell cycle marker like cyclin D1, proliferation marker like DDX3, Ki67, both at the transcriptome as well as the proteome level, and can be clearly seen that there is significant inhibition of cyclin D1, DDX3, Ki67, as well as VEGF in drug alone, as well as combination of ketorolac and sunitinib, clearly indicating that ketorolac alone as well as in combination would be able to inhibit migration and angiogenesis. Now, we also simultaneously were working on this very important tumor suppressor protein, prostate apoptosis response 4. And parallelly, what we did was we tried to find out whether ketorolac is able to modulate this particular tumor suppressor protein. It is very well known now that PAR4 is downregulated in numerous cancers, including renal cell carcinoma. PAR4 is a selective pro-apoptotic protein encoded by a PAR4 gene. How would be PAR4 a selective pro-apoptotic protein? There are two reasons for it being very selective pro-apoptotic protein, specifically causing apoptosis in cancer cell. Two mechanisms. Number one mechanism is the high level of endoplasmic reticulum stress. High level of protein kinase A in tumor cell leads to the specificity of PAR4 against cancer cells. Second specificity of PAR4 as a pro-apoptotic factor is that cancer cells are known to express high levels of GRP78. And it has been found out that GRP78 is the binding partner for PAR4. And these are two coveted features which provides PAR4 with its specific pro-apoptotic activity against cancer cells while sparing the normal cells. Ketorolac acts by two methods. The first method which with ketorolac can act is through an intrinsic or an intracellular mode. And the second method is it can act through a extracellular mode. Now, the picture on the left-hand side of the slide shows the intrinsic method or mechanism of action of PAR4. If the internal intracellular level of PAR4 is increased, now in cancer cells, I told you that the PAR4 levels are significantly downregulated. If by a method we are able to upregulate the amount of PAR4 intracellularly, and I'll come to that, how can we actually do the upregulation of PAR4, but if we are somehow able to upregulate the amount of PAR4 intracellularly, then PAR4 would bring about the apoptosis of the cancer cells via the caspase-3 pathway. At the same time, if we are not able to bring about an intrinsic or intracellular upregulation of PAR4, there is a second mechanism by which PAR4 can bring about that action, which is by stimulating the normal cells of the body to secrete PAR4 systemically. The diagram on the right-hand side shows a picture which was presented earlier by another group working in the University of Kentucky, where they have shown that a particular drug called as chloroquine is able to induce normal cells of the body to systemically release PAR4. The PAR4 released systemically then would be able to bind to the GRP78 markers that are present on the cancer cells. I told earlier that GRP78 is upregulated in cancer cells, and GRP78 is the binding partner for PAR4. So the PAR4 that is systemically released would bind to GRP78 specifically expressed on the cancer cells, and again bring about the apoptosis of cancer cells via the caspase-3 pathway. So these are very important feature of this novel tumor suppressor protein, that one, it is highly selective for bringing about apoptosis in cancer cells, and two, it has these two distinct mechanisms of action to bring about the anti-tumor activity, the intrinsic mechanism or intracellular mechanism, and the extrinsic mechanism. And I've already told you earlier that the level of PAR4 is decreased significantly in several cancers, including RCC. And our another work, we have shown that in patients of glioblastoma, of GBM, have very low level of systemic PAR4 as opposed to normal healthy volunteers. So therefore, we tried to find out if Ketorolac by itself or in combination is able to bring about upregulation of intracellular PAR4 in cancer cells, as well as able to upregulate or stimulate normal cells in order to secrete PAR4. So we did both. And here is the data. Just excuse me for 1 minute. Somehow my laptop is conking off. Just give me 1 minute. I'm so sorry about this. I'm so sorry about this. I hope that my screen is still visible, I'm audible, and I am also visible.
Yes.
I'm sorry. So, so this is the data that I wanted to now present, where we wanted to look at the ability of Ketorolac to upregulate PAR4 from normal cells as well as upregulate PAR4 in cancer cells. So the figure on the left-hand side is showing the ability of PAR4 to upregulate ability of Ketorolac to upregulate PAR4 from the normal cells, both at the transcript as well as the protein level. And with the increasing concentrations of Ketorolac, as you can see in the left-hand side picture, there is an increasing amount of PAR4, and then at both the transcriptome as well as at the protein level. So that's the normal fibroblast cells that we are talking about. At the same time, the top plots in the middle and the right-hand side shows the ability of Ketorolac to upregulate PAR4 in the cancer cells. So we have used A498 cells as our model cell line, and we have shown that it is able to upregulate PAR4 both at the transcript as well as the protein level. So this is a very important discovery that Ketorolac is capable of bringing about upregulation of PAR4 both in normal as well as in cancer cells. And that gave us that significant boost that yes, Ketorolac is going to be highly efficacious against RCC. We have also looked at other markers like TIM1, RAC1. Of course, it's being known that Ketorolac can inhibit RAC1. It's being known that Ketorolac inhibits CDC42. So we did that study specifically using RCC cell line A498 to look at whether the Ketorolac is able to cause downregulation of RAC1 and downregulation of CDC42 in A498 cells. That's something new, even though it's being known that it causes inhibition. But in RCC, this is the first time that someone is showing. And as you can see from these plots on the right-hand side, when we use the A498 cells, Ketorolac is not just able to inhibit the PAR4, TIM1, RAC1, CDC42 alone, but it also causes additive or synergistic effect when combined with sunitinib to bring about inhibition of PAR4, TIM1, RAC1, and CDC42. So it's going to work absolutely fine when it is combined with these standard of care drugs. In order to prove our point that Ketorolac does mediate its mechanism of action through upregulation of PAR4, we conducted siRNA-mediated knockdown studies in A498 cell lines. The figure on the top right corner, I hope that everyone can see the cursor that I am using. The first panel of blot shows the expression of PAR4 with siRNA-mediated knockdown of PAR4 at 2 hours, 5 hours, 18 hours, and 24 hours. And as we can see very clearly, starting from 5 hours, from 18 to 24, there is a significant knockdown of PAR4 levels post-siRNA knockdown. Then, if you look at the blots in the middle, which is talking about RAC1, it is clearly seen that if there is a downregulation of PAR4, it leads to significant level of upregulation of RAC1. So I will use, so if you see the 18-hour data, the PAR4 here, if you can see, is significantly downregulated, and the RAC1 is significantly upregulated. The densitometric, the values on the top are the densitometric analysis. So also in 24 hours, if you see this section over here, the PAR4 is significantly downregulated, whereas there is a significant upregulation of RAC1. And the reverse is true. If you look at the panel on the right-hand side, if we do a downregulation of RAC1, there is an upregulation of PAR4. If you look at, here we have downregulated RAC1 by siRNA-mediated knockdown. So here, the RAC1 is downregulated and the PAR4 becomes significantly upregulated. So there, with this data, we are saying that there's a definite direct crosstalk between PAR4 and RAC1. And RAC1 directly controls CDC42 and other proliferative, metastatic, and angiogenesis feature. So we are clearly showing for the first time that PAR4 and RAC1 crosstalk with each other. In RCC cases, there is a significant downregulation of PAR4. There would be a significant significant upregulation of RAC1. There would be therefore significant upregulation of CDC42, which would lead to proliferation, metastasis, angiogenesis. Now, why am I saying significant upregulation of CDC42? Because of the blot that is present over here. If you see this. Here we are showing the levels of CDC42 post PAR4 silencing and post RAC1 silencing. Now, if you see at 24 hours, if you see this particular lane over here, this is the RAC1 silenced cells where RAC1 silencing leads to a significant downregulation of CDC42, and RAC1 normalization would therefore lead to significant upregulation of CDC42. If you go to the blots on the right-hand side, we have looked at the effect of PAR4 and RAC1 silencing on the expression of VEGF. And we are clearly seeing over here that downregulation of PAR4 causes upregulation of VEGF. And this is the graph that I'm talking. This is the blot that I'm talking about. If you see at 24 hours, PAR4 silencing leads to significant upregulation of VEGF. So what we are showing for the first time is that if you upregulate PAR4, it will downregulate RAC1, which in turn would downregulate CDC42. There will be a downregulation of VEGF, and therefore we are saying that Ketorolac would be a very important drug to prevent metastasis as well as angiogenesis. So once we know that Ketorolac at the in vitro level, at 2D, 3D combination studies, mechanistically is able to bring about its efficacious action, we went to the next step to look at the ability of Ketorolac alone and in combination to bring about anti-tumor efficacy in a xenograft model. And the data, and we're very substantial and significant data that we got that Ketorolac alone as well as in combination with sunitinib is about to bring about significant anti-tumor activity. If you look at the graphical representation of tumor weight in terms of milligram, Ketorolac in combination with sunitinib, the red graph, the right-hand side top corner that I'm talking about, the red colored bar clearly indicates that Ketorolac significantly inhibits the tumor progression and proliferation in combination with sunitinib. We also did PD analysis from the tumors that we got from the study. And we are again clearly seeing that proliferation marker like DDX3, the hypoxia marker like HIF1, CDC42, of course, we looked at, RAC1 we have looked at, β-catenin marker, another proliferation marker that we have looked at, is significantly downregulated at the transcriptome level, alone as well as in combination with sunitinib group. So at each stage of our study, starting with in vitro 2-dimensional, 3-dimensional, in vitro 3D combination studies, the proteome analysis, genome analysis, the PAR4 expression level, the pathway that we have elucidated, the xenograph studies that we have conducted, each of these stages is clearly indicating that Ketorolac would be a great drug to be used in combination with current standard of care against renal cell carcinoma. Now, here I would like to bring about a very important point. We are showing it for the first time that Ketorolac is able to cause upregulation of PAR4 from the normal cells. And as I told you, PAR4 is downregulated, it has been shown to be downregulated in several cancers. So there is a very good potential that apart from renal cell carcinoma that we have shown for the first time, there is a possibility that Ketorolac, depending upon the mechanism of action of as a PAR4 secretagogue, would be able to be repurposed against other solid tumors also. So the role of Ketorolac may not just be restricted to one indication that we are showing over here, but it may also be repurposed for other indication given its new mechanism of action that we have elucidated for the first time. So what have we discussed till now is that what is drug repurposing, what is, why is it essential, how can drug repurposing be done? I've shown the preclinical proof of concept studies for Ketorolac against RCC, where we discussed about in vitro profiling, the migration inhibition, anti-angiogenesis potential of Ketorolac, the biomarker expression specifically related to TIM1, RAC1, CDC42, VEGF. We also discussed about the novel mechanism of action of Ketorolac, and I also showed you the in vivo efficacy studies in the xenograft model for Ketorolac alone and in combination with sunitinib. So on the basis of the studies that we have conducted, this is the mechanistic action that we have proposed for Ketorolac against RCC. Ketorolac directly causes inhibition of TIM1, RAC1, CDC42, as well as via the upregulation of PAR4. Upregulation of PAR4, on the other hand, would directly be related to inhibition of HIF1, VEGF, and other proliferation markers. So, all in all, what we are saying is Ketorolac is a wonderful drug which could be repurposed against renal cell carcinoma. Coming up to this point, what could be the potential next step? Now that we have proved it till this level that Ketorolac is a wonderful drug which could be repurposed for renal cell carcinoma, the next step, therefore, being a pre-approved drug, to conduct a clinical POC studies in a smaller population to show its efficacy. As an organization, we are now in the process of doing that. We are also in the process of communicating with the FDA as a pre-IND submission to get the feedback from the FDA. And of course, we are also looking at what kind of anti-generic strategies can be used in order to get some amount of exclusivity and get some amount of advantage in terms of business out of the studies that we have conducted. So thank you again for this wonderful opportunity. Thanks to all the organizers of ICC, from all the authors, along with me, Dr. Vinay Sanavne, Dr. Swati Atsrekar, and Dr. Kalpana Joshi were an integral part of this entire program. Thank you very much, and I would be glad to answer any questions or queries that the audience would have.